especially throughout the Southeast. You'll see Jim and members of his team there, and so we'd like to just thank him for his support. Everything that's done. And with that today, he's I'm sure I'll let him really do the full justice, but it's going to be deep diving into the science of the microbiome. And we're learning each and every day that the microbiome is central to our health for so many ways. And microbiome is central to our health for so many ways. And I'm excited for all of us to learn just a little bit more about the science and the research that goes into that. And so with that, I'll turn it over to Mr. Jim Daly.
Okay, thank you. Thank you.
Thanks, Jim.
And well, it's nice to see you here. And we're going to talk today. Let me get this thing going. Oh, okay, I got to get to the other one. Oh, okay, I got to get to the other one. Okay. Okay, where are my slides? Okay, there we go. Okay, we're going to talk about understanding the human microbiome, and really particularly the gut microbiome, because when you talk about the human microbiome, that is more than just the gut that we usually think about. Actually, we have microbiome on our skin, on our head, in our mouth, all over us. Our whole body is has bacteria that forms a microbiome. However, what we really deal with most of the time is the gut and the intestinal tract. Now, there's some terminology that we'll use. First of all, probiotic, when we talk about probiotics, we're talking about the live bacteria. So I think everybody understands that. When we talk about prebiotics, depends on how you define it. That's not a carefully defined term yet. Some people will consider a prebiotic as something that's a food portion that's not digested and goes into the gut and is digested and metabolized by the gut bacteria. And those are prebiotics. However, some people expand that to just about anything that goes into the gut and affects the microbiome there in a positive way as a prebiotic. So it depends on how narrowly or how broadly you want to define it. Many things have prebiotic effects. We usually think about things like inulin, the different types of fibers as prebiotics, but also things like the polyphenols are prebiotics as well, because they affect the gut microbiome and the rest of the body in a very positive way. And the microbiome actually modifies those and affects how they work in the body. Symbiotics are simply a combination of a prebiotic and a probiotic. So they work together. Then we talk about a postbiotic. Now that's something that's really interesting, and it's a fairly new term. A postbiotic comes from killed bacteria. Bacteria in your gut produce a lot of small molecules, and one of the ways they work is by releasing those into the gut. But you can kill the bacteria, take an extract of it, and get a lot of the benefits of the bacteria just by supplementing a person with those active components. And that's getting to be a fairly hot research area. And they're being starting to be sold as, sometimes they call them prebiotics, sometimes they call them postbiotics, but they're a prebiotic that comes from a probiotic that's been killed in some way, and just what's left of it. We also talk about symbiosis. That's where the organisms benefit each other. Most of the what we call probiotics are symbiotics also because they benefit uh, the host, us. Um, Commensalism, that's where the bacteria just sort of live there and don't benefit you or hurt you or anything. They're just there. So that's another way. Amphibiosis is two life forms that can may harm or help each other depending on the conditions. Now that's a pretty common situation in the gut. Actually, there's a lot of bacteria like that, and we'll talk about some of them. And then when we're talking about bacteria, a lot of times, if we're talking about the dose, the strength, colony forming units is what we'll be using as the measuring tool for that. And that's comes from when you do an assay with live bacteria culture. You basically dilute it and dilute it down until you can get that on a plate, and you'll see little dots where the bacteria are growing. And that will tell you how many bacteria are in that concentration of media. So that's just a way of measuring the amount of bacteria. And that describes the method that we use for that. And here's one little ad, so to speak. This is a book that just came out that I'm a chapter author on. Turns out I'm the only non-Korean author in that book. But I edit the journal Medicinal Food for the Korean Society of Food Science and Nutrition. And I do a lot of research with Koreans. So I got involved with that book. All the editors there, the four, are good friends of mine. So I got a job of writing about Jang, which is like natto, the equivalent to natto that Koreans have. So that's a very good book, though. There's a lot of chapters other than mine about fermented foods. Koreans eat a lot of fermented food, and those are very important probiotic foods, postbiotic foods, prebiotic foods, all of it. So they're really great foods for your microbiome. And if you really want to get into the science of those kind of foods, that's a very good book from CRC Press. You can get it on Amazon or wherever. Now, we're going to sort of back up a little bit because I want to bring into focus the human microbiome from a historical context as well as a scientific one, because we've been involved as humans with bacteria for a long time. The very early people were aware of the effects of bacteria, but they didn't realize what they were, what was doing it. But they realized that food would go bad and it would make them sick. So they cooked their food, they froze their food, they smoked their food, dried it, they ate it raw when it was fresh. But they treated it in ways to preserve it, not knowing why it needed it, but they just knew it worked. Now, further down the road, fermentation came into the process. And long time ago, in probably a land very far from here, I kind of visualize a lonely traveler who was kind of walking through the mountains there and found good food to eat, vegetables out there. And so he ate all he could. He was full. He wrapped it up in a skin and buried it somewhere and thought, I'll come back and have some more to eat later when I come back through. But it probably took him longer than he thought. And he ended up discovering fermentation. And this is an old Korean type place. And what he probably had was something that was very similar to the Korean kimchi, which is kind of the signature food of Korea, if you all know about it. It's actually a wonderful food. It's strong, but you talk about some health benefits. We've got these cruciferous vegetables that have been fermented with peppers and garlics and all these wonderful bioactive compounds. It's pungent. It's hot. But it doesn't have to be actually. Kimchi can be made very mild. Sometimes they don't put the pepper. They make what they call white kimchi. It's just fermented cabbage and a few other little things, some radishes and things like that. So it doesn't have to be hot, but often it is. But you've got a lot of live bacteria in kimchi, and you've got all these prebiotic vegetables. You've got so many things, and if it's uh, traditionally fermented, like much of it is, it's got a wide range of bacteria in it. And fermented foods actually have developed all through the world, and you can just kind of see a scattering of them there. And these foods all developed well before people knew anything about bacteria. They just learned these methods by accident. And it happened everywhere. The funny thing is, where it didn't happen was in the Americas very much. Very little fermented food, actually, in the Native American, South American, or North American foods. So somehow this fermentation culture didn't uh, come over across the Bering Straits, I guess, with the uh, early Americans because it just wasn't a major part of the Native American food. Now, we'll just keep moving in time. This really, we're getting to fairly modern times when Leeuwenhoek invented the microscope essentially and became the first person really to become aware of the microbial world. Uh, because when he got his microscope to work, he saw all these little things, animals moving around there. Called them animales, something like that. Uh, Moving through time, Louis Pasteur was really the first to discover that those bacteria cause fermentation. Uh, and very clearly showed that they didn't come about as spontaneous generation, which was a major breakthrough. He also established a link to microbes as a cause of disease and invented pasteurization and immunization. He died at age 72. Leeuwenhoek died at age 90. And you hear people say people didn't live very long back in those days. A lot of them did, actually. uh, yeah, Leeuwenhoek died at 90, Pasteur at uh, 72. And then there was Meshnikov, which really was the father of probiotics. He was an immunologist, uh, was a contemporary really with Pasteur, died at 71, and he postulated that the bacteria in yogurt might be responsible for the long lifespan of people in Bulgaria. And he gave a lecture about that in Paris. And he was kind of a celebrity scientist at the time. And people just went nuts after that. And the man for yogurt just skyrocketed in Paris, and they couldn't keep it. And really, that's what really has resulted in the popularity of yogurt today. He really started that. And he was the first really to show a probiotic beneficial effect of the microbes. Everybody else, well, they could be used to ferment food and preserve it, but not really a health benefit. He was the first to come up with that idea, really, and or at least to popularize it. Now, a few years ago, I was really blown away by this guy's research. O'Keefe, as a medical doctor at University of Pennsylvania, I believe. And I was at an experimental biology meeting in Chicago. And he showed this research where he was studying African Americans who get colon cancer at a pretty high rate. And he had a specific group whose ancestry he could trace back to a particular region of Africa. And those people in Africa didn't get colon cancer. And so he was wondering, well, why? So he decided probably it was linked to the diet. So he took the African Americans in his area, the group he had, and the people in Africa, and he went to Africa and worked with them there, and he switched their diets completely. Now, the people in Africa, their major diet was cornmeal, a coarse cornmeal that they hand ground uh, with all the bran and everything in it. Uh, and that was, you know, by far the major calorie source for those people. Um, Whereas the African Americans, they ate a lot of meat. They were very heavy meat eaters and, ate pretty much a standard American diet. Well, he reversed the diets. And the fiber switch was quite a lot. The African usually got about 55 grams of fiber per day, where the Americans got 14 grams of fiber per day. Very big difference. The Africans only got about 16% fat in their diet, whereas Americans got 35% fat in their diet. So he switched them. And he actually did colonoscopies and took samples and looked at how the colon changed. And sure enough, within 6 weeks' time, the Africans in Africa were developing the precancerous polyps and lesions already that were characteristic of the Americans. They were disappearing in the Americans. Now, they didn't continue the study much longer because you don't want to go to Africa and give everybody colon cancer. And they were very happy to die. They don't eat the diet of just corn because they love corn so much. It's cheap food is the reason they eat it. They loved me. They enjoyed that. They was great. They brought all those steaks and stuff from America for them. They were, thrilled to death. But literally. But no, it was. He showed the slides of the biopsies and things, and it was really pretty amazing the difference that made in those people on both sides. So we very clearly had an effect of diet, and it was traceable to the microbiome because there was just a huge change in that amount of time in the microbiomes of the two groups. One of the major bacteria that were affected was the bacteria Akkermansia muciniphila. Akkermansia is one of the major bacteria that were decreased by the high meat diet. It's also depleted in obese people, and it's associated with leaky gut syndrome. What it does is eats mucin that lines the gut, allowing for more of it to be made and to maintain that mucus layer in the gut. That's how it actually works. You have goblet cells that make mucin all along your intestinal tract, and that forms a barrier that protects the gut. Also makes a place for good bacteria to colonize and stay there. So it's a very beneficial, mucin is. However, it has to be recycled, and that's what the Akkermansia does. It keeps the old stuff leaving, and it metabolizes it, and then the new stuff is being made. And when that's in balance, that's great. It works out very well. Too much is also bad, however. You can have an overgrowth of Akkermansia and cause problems. As it turns out, people with inflammatory bowel diseases, colitis and Crohn's disease, they often have too much of it. So, it's one of these, like we were talking about earlier, that you can, depending on the situation, it can be good for you or bad for you. It's getting the balance. And this is kind of the trick to understanding the microbiome completely. The concept of good bacteria and bad bacteria is not always correct. It's really a balance of them. because many of these bacteria, and this is one of the examples, can be both good and bad depending on how much you have of it and where it is. Did you have a question?
Is this induces IL-10
Um, it's, you know, it is, if you, the people who don't have it adequate and are obese, they do tend to be more inflammatory. I'm not sure that it produces IL-10.
studies 20 years ago, I used lactobacillus plantarum that induces IL-10 inflammation.
Mm-hmm. Okay. Okay. Another study just recently, and that's the title of the study, it was published in the journal Nature. Actually, not real recently, a while ago, studied that switching to either a very heavy meat diet or a plant-based diet really changes your gut microbiome very, very rapidly. We're talking about 2 or 3 days, you see big changes in the gut microbiome. Most of the time, most people's microbiomes stay very, very same throughout many years because tend to eat the same diets. And so microbiomes, unless you take a lot of antibiotics or something, usually are pretty stable for a long time, but they don't have to be. Um, they found that it can rapidly change depending on your diet, and they took and switched people on groups between a plant-based diet and a meat diet. They found that the high meat diet greatly increased biophilia and decreased firmicutes. Now, biophilia, these are um, bacteria that are tolerant of bile acids. And most bacteria won't live in the small intestine, particularly because of the bile that will destroy them. But these particular bacteria can stand that. Well, when you eat a lot of meat, a lot of fat, you produce a lot of biles, and that encourages the growth of the biophilia. And so that's turns out that the biophilia are very highly inflammatory bacteria. So those are ones you really don't want to overdo. Some are probably good. Uh, but the results in that study were quite profound. And David Lawrence, one of the authors, said, and quoting NPR, actually, he was talking to them about the study. He said, I mean, I love meat, but I will say that I definitely feel a lot more guilty ordering a hamburger since doing this work because he showed that it really, the plant-based diet was just so much healthier for the microbiome. It was just no comparison in that study, just like in the African study that we had. So these plant-based diets are very clearly um, what you need for the microbiome. Now, interestingly, and I don't know quite how to attribute this, but we hear a lot these days about inflammatory bowel diseases, Crohn's disease, and ulcerative colitis. Well, look, before 1990, there was almost no ulcerative colitis in this country or in the world. If you go back as far as the 1960s, there weren't any. It hadn't even been described yet. It was very, very rare. And just in recent years, it skyrocketed. And this is CDC data, and what do we attribute that to? Well, we're not quite sure, but it probably has something to do with the microbiome. Uh, Fried food, maybe. Antibiotics. Um, mhm. Yeah, it's hard to get people to change their diets. There's no doubt about it. Very difficult. But um, it might be attributed to antibiotics because look at we didn't really have antibiotics until 1943, I think, is when penicillin really came on the market, and it started growing from there. So antibiotics, a lot of research has shown in epidemiological studies that the antibiotic use, looking at different countries, really can be traced back to the microbiomes of the countries. Antibiotic use is a major, major factor in uh, gut diseases and dysbiosis, which means, a disturbed microbiome, basically. So it's very likely that antibiotics are really what has brought this on, but we don't know that for sure because there's no way really to prove it or disprove it.
like the like lactose, xanthan, and other ones that make you produce B complex.
Yeah,
But if you look at it, most people's breakfast, what's in the morning, coffee.
yeah,
Coffee will deplete all your B complex,
okay,
including your lithium, thyroid, and the lithium. They had studies a couple years back, this back to 2013 in the Crohn's conference, Penn State University, the Foster study. The lithium it regenerates the colonic cells. But nobody knows what depletes the lithium, the coffee and the salt. This is very highly used for everybody, but when we don't know who is the enemy, how do you fight it?
yeah,
You can't fight it.
sure. Well, let's just go back and look. When we're born, we have an almost sterile, not quite really. They're finding out that it wasn't true. They used to say that infants had sterile guts. They don't really, but they come pretty close. There's not many bacteria. So during the first 2 years of life is when you really start to get some population of bacteria that amounts to anything. And you can see just like at birth, 1 year old, you're starting to get some people in there. By preschool, you've got a whole city full of bacteria. And then what happens? We take antibiotics, and where do we go? Well, back here again. You can literally almost do that. That's a little bit of an exaggeration. But the antibiotics can really wipe out a lot of bacteria.
Also, during the birth, the vagina is a special bacteria, but a lot of people they do cesarean sections, and also if you know longer before I used to that comes in the breast milk, but how many people they do the breast milk?
Yeah.
So everything is limited, but alcohol, total alcohol is dead bacteria.
Hmm.
So
That's
we
true.
replace the dead bacteria with their life, and the same thing with the bread, the raise the dough, they use dead bacteria.
Yeah. Okay. Well, what we're finding though is that bacteria really grow rapidly. And we've seen this in some of the studies we've talked about before, how the bacteria change very quickly when you change diet. And this is sort of a growth curve that you would see if you made yogurt. Uh, you can see that over time, a lot of these bacteria will double in counts every 20 minutes. That's very common. Some of them faster than that. So uh, then it will go up to a stationary phase and then drop off. That's your typical bacteria growth curve. Um, in the human body, it's not quite like this. This would be more like I said, you would see in yogurt where you have one bacteria and you're culturing it. And in yogurt, the lactic acid it produces really builds up to the point it becomes toxic to itself. And then they start declining. In the microbiome, you have a different situation. You have that explosive growth if the conditions are right, but then it's sort of held at bay by other bacteria who will metabolize some of those things that it's producing so they can sort of level out and be more stable. Now, we have a lot of different, I'm going to kind of move on a little bit quickly here in in the gut. And this is illustration I made that shows the taxonomy of what lives in the gut. And it's not just bacteria. If you look up the domain we have three types of life forms. Archaea are very primitive. People used to think they were bacteria, but they're not. Actually, they're closer to us than they are to the bacteria, but they're very small still. Um, but those life forms can grow in the gut and a lot of other places. The bacteria, which we usually think about growing in our guts. Also, under eukarya, the eukaryotes, we have the fungi, the yeast, and things like that. And those can be important microbes in the gut as well. Now, with the bacteria, we just kind of go through the phylum. You can see that the phylum, we have really 5 bacteria phylums that are common in the human gut. The eubacteria, they're all under the eubacteria, but the fusobacteria, proteobacteria, actinobacteria, the Firmicutes and Bacteroides. Now, these two right here, Firmicutes and Bacteroides, those two categories form 95 to up to 99% of the bacteria in the human gut microbiome. Those two categories are responsible for most of it, although there's some important ones that are actinobacteria and proteobacteria, but they're in much smaller quantities. So when we go under Firmicutes, you can see we go down to the class, order, and family. We can finally get over here to Lactobacillus acidophilus and even go down to the strain, DDS1. So these are very important things to consider. First of all, the strains often are just as important as the species themselves. If you look at E. coli, E. coli can actually be fairly beneficial under certain circumstances. It can produce vitamin B12, although we don't absorb the B12 that it produces. Unfortunately, where it's produced is in the large intestine, and it can't get back up to the small intestine to be absorbed, but it can make it. Well, it can't be absorbed there in the gut, in the large intestine. It's very hard to absorb vitamin B12. And it doesn't appear that it can be absorbed from there, even though there's a lot of it in there. Biotin is another matter. It's also produced by the bacteria, and it can be absorbed some in the gut. Yeah. Vitamin K is made by these bacteria, so they can be absorbed. But it depends on the DDS1 is the acidophilus strain that I like because it's one that is pretty stable and works well. But the Firmicutes and Bacteroides, when you start reading the literature, you'll see these compared a lot. And I encourage you to read the literature some because they'll often talk about how different prebiotics or different treatments affect the ratios of those. Generally, Firmicutes is a little bit better one to have more of, I think, than the Bacteroides, but not necessarily because there's good bacteria in both of them. So just saying that increasing one is better than the other, I don't quite buy that, but you'll hear that quite a bit. Now, the Archaea really are ones that interest me a lot, and we don't know a lot about them, but you hear about these microbes in Yellowstone that live in the hot springs. Those are Archaea. They're not bacteria. And they can live at literally right at boiling temperatures in very caustic conditions. They're really amazing. They're the ones that can literally eat plastic that's out in the ocean. It's a slow process, but they can do it. They can eat the petroleum from oil spills. They're again a slow process, but people are wanting to figure out if they can actually find a way to use these to do it in a practical sense. They live in the thermal vents down under the ocean. So these are really an interesting category of microbes that I really hope to be able to learn a lot more about. But the research is really in the infant stage for those. But they have the potential to detoxify a lot of things. And that's what really interests me is perhaps some of the Archaea, if we learn which ones and how to use them, have the potential for really getting rid of a lot of toxins in the intestinal tract. Um, from accidental poisoning or just from foods. I think the potential is there, but we really haven't gotten there yet.
How hard is it to reproduce them?
What's that?
How hard is it to get them to reproduce and grow?
Some of them aren't too bad. Some of them are pretty hard. Unfortunately, the reason they've had a hard time with reproducing a lot of them is because they live in extreme conditions, and they have to live in those extreme conditions. You can't culture them in normal lab conditions. You can't. It's hard to. You could, you know, uh, in the lab, create a thermal vent type situation by heating up a flask of water and put in a lot of the caustic sulfur materials and things like that. You could do it, but under normal conditions, they just don't grow. They have to have these extreme conditions to survive, the ones that live in those kind of situations. The ones that metabolize plastic and oil, they can live in culturable conditions. So there is some research going on those. It just depends on which kind they are. But they so far haven't been ones that have been really studied as nearly as broadly as the bacteria. And that goes back to one of the reasons why they've always studied bacteria, because most of us have grown up thinking, bacteria are evil things we've got to get rid of. We need all these antibiotics, we need all these antiseptics, we need all the, antibacterial soaps and all this kind of thing. And actually, the antibacterial soaps may not be that good. There's a microbiologist the other day talking about this on NPR. He was telling us, the audience, that when you're taking those antimicrobial soaps, he says you're really doing more harm than good. You're better off with just plain soap. The reason being, the bad bacteria that really are nasty are not touched by those antibacterial agents they're putting in there. Uh, and so what you're doing is you're getting rid of most of the bacteria, but leaving the really bad ones alone. And you're sort of selecting for those really nasty bacteria. So, yeah, so maybe you're better off not getting the antibacterial soap, according to this microbiologist anyway, and just using plain old soap and water for cleaning.
After that, you put your hand on the microscope, so micro,
Yeah.
not clear.
Well, some of these bacteria are pretty, resistant. Like Pseudomonas bacteria is really nasty stuff.
It's very important.
But
Brain weighs 3.5 kilogram and gut bacteria weighs 3 kilogram.
yeah, but Pseudomonas can live in alcohol. It metabolizes it. So you're using alcohol, rubbing alcohol or something to sterilize stuff. It's not going to work on the Pseudomonas because it'll just eat it and grow. So you got to, consider those things sometimes. So how do microbes benefit humans? And this is one of the slides I really wanted to talk about. We hear a lot about this. This bacteria is good for this condition. This bacteria is good for that condition. But how do they actually do it? What do they do? And so let's just take a look at that for a minute. First of all, one of the most important things that particularly um, the Firmicutes do is produce the short chain fatty acids, acetate, butyrate, and propionate. Now, those short chain fatty acids do two things, well, two categories of things. First of all, they feed the cells in the gut. That's the food that helps the goblet cells, epithelial cells, all those to live, because they depend on that for their food. So to have a healthy uh, endothelial population in your gut, they need those short chain fatty acids. That's their energy source. The second thing they do is actually they are a signaling molecule that can be absorbed out of the gut and send signals around the body. And that's something that's becoming a very interesting thing. Some of these acetates and butyrates actually travel through the bloodstream and go to the brain and other places and are signaling molecules. One group of bacteria actually even metabolizes tryptophan. And I actually had that paper here, I think. Yeah. This was in the journal Nature, which is a very, high-level journal. It's Nature and Science are the two top scientific journals in the world, and they almost never have nutrition research in them. But they've been publishing quite a bit about the microbiome, and that's really unusual because they are getting into our areas in these really high-level journals. But they found that microglial control of astrocytes is controlled by microbial metabolites, and particularly metabolites of tryptophan that are being released from the gut, going through circulation, being taken up across the blood-brain barrier, and is really having an anti-inflammatory effect in the brain, protecting the brain against diseases like Parkinson's disease, Alzheimer's disease, encephalitis type diseases, those things. And that's a major breakthrough. You're welcome to look at that. But very protective effect of the microbiome on the brain and those kind of diseases. The microbes can change bioactive compounds in foods to make them more effective. And this is getting in more in my area. I have a paper coming out in the Journal of Medicinal Food in about 3 months where we looked at how gut bacteria change an isoflavone that's found in soybeans. It can be found in kudzu root, it can be found in red clover, a lot of Shingman, come in. So it can be found in a lot of places, but it takes diadzine that and converts it to equal. Now, equal is a very powerful, compared to the diadzine, signaling molecule in the body, and it is what really makes isoflavones work in women, as far as for premenstrual syndrome, for menopause symptoms, things like that. And we did a meta-analysis of a lot of different clinical trials and found that really consistently women who had the right bacteria to make equal had much, much less symptoms from menopause than the women who didn't. It made a really profound difference. And that'll be coming out. It's already accepted, and it's going to be about February or March, it'll be published. So, but those kind of changes, that's just one example how the microbiome can affect various polyphenols and other things in the body to actually change how they work in the body. A lot of times in the literature, you'll see where, they say, well, these polyphenols, well, curcumin or the isoflavones aren't well absorbed. How can they do anything? You give these to people, you measure it in the blood, you don't see that much going there. How could they be effective? And yet the clinical research is showing it's being effective. Well, one of the reasons is a lot of times they're not the same molecule anymore. The gut microbiome has changed them into something else. That's true of curcumin. Curcumin shows up all through the bloodstream if you take and put label it with a radioactive tracer and give it to mice or something. You don't do that to people, but but it shows up as a whole bunch of different compounds throughout the body. it's not curcumin anymore. It's different things that the curcumin has been changed to. That's in there working. And that's why, even though maybe the curcumin isn't that well absorbed as curcumin itself, it may be still very effective because of how the microbiome changes it. The microbiome produces immune factors, which improves tolerance to antigens. It can make some essential nutrients. We mentioned vitamin K and biotin. We know it makes vitamin B12, but we're pretty sure it doesn't do us any good because of where it's made. It literally produces neurotransmitters, the microbiome does. And the gut-brain connection is very, very big right now in the research. And it helps with maintaining body weight, maintains benefits for cardiovascular health and blood glucose regulation. And I've got a number of papers showing all of those effects. So we can see the microbiome does a lot of things throughout the entire body, the effects of it. The most studied of all the quote probiotic bacteria is probably Lactobacillus acidophilus. Actually, that's the one that Meshnikov that we talked about earlier, one of the ones that he discovered and began working with was the acidophilus. It's a gram-positive anaerobe, but does tolerate oxygen pretty much. Surprisingly, it does pretty well throughout the body. Actually, you can find it in the mouth and in the throat in small amounts, quite a bit in the small intestine, not quite as much as in the large intestine, surprisingly. It doesn't show up as quite as much there, but it's probably the most important bacteria in the small intestine. Like most good bacteria, it probably helps by reducing inflammation by encouraging the T regulatory cells to produce IL-10, which you were mentioning earlier. And also, it produces bacteriocins that can kill other bacteria, often quite specific for the more what we consider harmful bacteria. So the bacteriocins, and that's really actually a characteristic of all the lactobacillus category of bacteria. All of them are produced to one extent or another, these bacteriocins that can diminish other bacteria in the gut. And often it's more potent against what we consider the bad bacteria. I sort of hate to use that phrase because a lot of these bacteria, like I say, are both good and bad, as we mentioned earlier. And a real example of that is the Helicobacter pylori. We usually think of that as really a nasty bacteria. It's known to cause ulcers. In fact, just about any time a person has a stomach ulcer, it's due to an overgrowth of Helicobacter pylori. Now, Martin Blaser, who wrote the book Missing Microbes, writes in that book how he was treated for ulcers with antibiotics. And they gave him a pretty stiff round of antibiotics and literally completely wiped out the H. pylori in his stomach. I mean, it was gone, period. And you don't have to have it, but then he noticed that he developed acid reflux disease. And he said, well, what's going on here? And is this just a fluke? It just sort of happened. And he started looking at the literature, and turns out people with acid reflux are twice as likely not to have H. pylori in their stomach. So H. pylori is probably one of the most demonized bacteria you can have. And yet it does, when it's under control, it does serve a useful purpose, apparently. And, that's sort of an interesting thing, but it really is typical of the whole bacterial complex that we have. Did you?
Actual immunoglobulin that have buffer the struggle and sustain personal immune system, and also the coffee and alcohol, it will make you secrete hydrochloric acid, and it will
Yeah.
So of course,
Hmm.
once you start drinking alcohol and coffee, no rest is replaced because there's no protein. I call it the fake protein because all the time when you eat protein, it's supposed to secrete hydrochloric acid and the pH is too very low. So actually, it also lowers your immune system because during the daytime, you have more glutathione in the stomach than the liver. It's going to strip that to an mucosal lining. And it's a win-win situation, but people that take proton pump inhibitors, so on and so forth. But when you don't look at where the real cause is coming, it's again, you just change your going nowhere.
Yeah. Okay. Well, I wanted to bring out two recent negative studies that came out on probiotics, and let's talk a little bit about that and why they had the results they did and what we can learn from them. Because sometimes the kind of like negative studies, we really hate to hear them a lot of times. It's like, but we can also learn from them a lot of times. And so, you know, I look at it as a way of fine tuning how we do what we do as far as helping people with their diets and their programs. So I don't look at them as negative as much as, helping us, fine tune our methods. The first one over here was in clinical gastroenterology, and it reported brain fogginess, bloating, gas, lactic acid acidosis that was more common among subjects using probiotics and linked to small intestinal bacterial overgrowth. And symptoms were alleviated after the probiotics discontinued. Okay, so people were taking probiotics, they were getting gas, bloating, um, and small intestinal.
They can be from the good bacteria, it's from the bad bacteria. They just study with the inulin. It's supposed to be the prebiotic. So the people with Crohn's use the inulin, my experience. It made them really gas machine.
Well,
And last year in the conference, Penn State University, the poster said they said, well, we had the exact same results you had. It's not a prebiotic, it's because if you have the pockets and Crohn's and colitis, I said, well, you can use them as a diagnostic tool if you take this and you have a lot of gas, then you have Crohn's.
yeah.
In fact,
Well, these people didn't have those diseases, um, but they were taking what we would consider good bacteria. But stop and think about it. There's very little bacteria comparatively in the small intestine. But when you take probiotics, they can't go directly to the large intestine where you're wanting them to go. They have to go through the small intestine. So if you think about it, it's no surprise, you know, that you would suddenly have a lot of bacteria growing in the small intestine if you're taking a good strong live bacteria. And I think a lot of people experience this with taking probiotics sometimes, is on the way to getting to the large intestine, it goes through the small intestine, which isn't supposed to have very much of any very high concentrations of any bacteria, pretty low. So you're going to have that happen, I think. I think that should not be a surprise. So as long as this doesn't continue for a long time, it's not something to really worry about. Uh, Essentially, it's just a matter of it's got to get to where it's going. And in the meantime, it's going to grow because that's what bacteria do. So I wasn't too concerned about that particular study. You don't want to have the uh, small intestine bacterial overgrowth be something chronic that lasts a long time. But it's not likely to be with probiotics because they don't tolerate the bile that's in the small intestine and the conditions in the small intestine very well. So that it's probably going to be a short-term thing while you're taking it. Now, if you were taking high doses uh, of probiotics all the time, you might, have a continual thing like that, and that's probably not good. You probably need to back off if you're having that problem. Now, the other one here was a little bit more worrisome to me. And this was published in the journal Cell, very good journal. And I've got that paper there if you want to look at it. They were giving probiotics, a commercial brand, to patients and to mice. They did it in humans and animals, tried both. After a round of antibiotics, it pretty well wiped up the bacteria. So then they looked to see how quickly after the round of antibiotics, the bacteria got back to normal. So back to where it started from. And usually they'll get there pretty quickly and pretty well over a few weeks at least, even if you don't do anything. So what they found, though, is that the bacteria in the stools took much longer to get back to, quote, normal in the subjects given probiotics. And this was a probiotic with about 11 different bacteria, a lot of them lactobacilli, and 2 tablets, each with 25 billion counts. So they were getting 50 billion a day in divided dose of this blend of probiotics. And the recovery to quote normal was much slower in those people and animals, mice both. Well, why would that be? Fecal transplants accelerated the recovery. And are you all familiar with what that is? I think everybody probably is. The fecal transplants, okay, where they give an enema of a healthy stool, essentially. So that worked well for returning the bacteria back to normal. So the consequences are not clear. First of all, one question I had though was returning the bacteria back to pre state before the antibiotics. Well, was that really good? Did they have that good of a microbiome to start with? So getting back to that, is that a good thing or a bad thing? Well, not quite sure about that. The other thing, though, that may be more important is when you look at the microbiome, you're looking at, in humans, somewhere between 500 to 1,000 different species of bacteria. Okay? Now, what did we say about the lactobacillus? They produce things that kill other bacteria, right? After giving people antibiotics, you've reduced a lot of the bacteria to very low levels. You may have wiped some out entirely, actually. Sometimes you do. But then those bacteria, the few that are kind of hanging on, you're hitting them with a lot of bacteria that kill other bacteria. They're good bacteria, and they kill bad bacteria a lot of times, but they're not that specific. Maybe that's not the time to give a high dose of lactobacilli, because you may be causing some of these bacteria that have already been very, very depleted to be further depleted by, quote, good bacteria. So that's what I'm taking away from that. If you're going to give probiotics after antibiotics, I think you want to give a low dose of it because you really risk getting rid of a lot of bacteria. And one of the things that you really want to do with the microbiome is maintain diversity. Most of those bacteria in your microbiome have never been cultured outside of a human or an animal. Probiotics make up, probably about 2% of those bacteria. So they're not really, there's a lot of good bacteria we just can't give people, but we don't want to destroy them either. So we have to be a little bit careful at that after antibiotics, I think, how we use probiotics to return them to normal, because I think that's what was happening. That's why it was taking them so long to return to normals, because they were giving them quite a lot of about five or six different lactobacilli that could suppress a lot of those other bacteria.
The one kind, actually, the DDS guy,
Uh-huh.
what's his last name?
The Lactobacillus acidophilus DDS1. Uh-huh.
But the guy who created it, he was a chemist too.
Oh, it was Shahani.
Yeah.
Uh-huh.
So in this book, he said, don't use multiple, just stick with one.
Yeah.
Multiple is not good. It's not like you are taking a B complex, you get everything, you know. So that's why Dr. Walford, 20 years ago, he studied on the lactobacillus plantarum. There's one thing, and it has results. And I used it at 50 million, and no gas, no bloating, except if the people that took the Inuit, and that made it worse. They feed
Yeah.
bad bacteria and stuff, probiotics, but the
Yeah.
single dose is better.
Yeah.
It's too bad they couldn't do a fecal sample for the antibiotic series and verify that it's a good microbiome, and after they're done with that treatment.
You know, I think they did, but they haven't really defined well what a good microbiome is yet. I mean, there's surprising diversity in people's microbiomes. And unless they really have a clinical dysbiosis, you really don't know very well how to. I mean, you're looking at these PCR information, DNA information, and you've got literally, like probably 600 species, and then you've got strains. Species is about 3% of the genetic material difference. And then you've got all of these strains in there, and it gets to be pretty complex. And they haven't yet figured out what a good microbiome looks like. To be honest, they really haven't. Determining core bacteria, well, I'll talk a little bit more about that in a minute, has been an area of pretty intense research. Well, people are asking that question, what does a good microbiome look like? And they haven't actually answered that question yet.
But the other question is, what are the bad bacteria and how the sugars feed those bad bacteria?
Yeah, well, we know that some bacteria are bad, but we also know that a lot of what we have always considered bad bacteria can have benefits. So you just got to keep them under control.
There'll be a lot of gas and blood.
Yeah. So anyway, that shows that we still have some to learn, and we need to learn how to use these things skillfully and not just, throw the kitchen sink at somebody necessarily. So, yeah, and this is what we were talking about, the core bacteria. Well, what do they look like? And, as we said, most of the bacteria belong to Firmicutes or Bacteroides and a few in the Actinobacteria, but that's only about 2 or 3% at the most, probably less of the Actinobacteria. Although some very important bacteria are in that group, the Bifidobacteria are part of the Actinobacteria. So we know that they're very important. But core bacteria, one study that was looking at this decided to look at the bacteria concentration in super old population, people that lived to 105 and older, assuming that they must have a good core bacteria. So what did they have? That was their approach. Akkermansia, they had quite a bit of bifidobacterium, and I'm not sure how to say it. Christensenale. I don't know. Anyway, that one's not one I'm really familiar with, actually. Akkermansia and bifidobacteria, but those were sort of the predominant ones. So they decided, well, that should probably be considered part of the core at least. The only ones that you can really culture and give as probiotics right now are the bifidobacteria. What's that?
The first one I used first, the Probiotic one, this comes from breast.
Uh-huh.
The one I'm using now, the top one, lactobacillus plantar, is the one I have that information. And also TNF too.
Uh-huh.
They get this other study, poster study, people with too much TNF alpha, were necrosis factors, simply they were short in biotin. Who knew that?
Yeah.
So you have to listen to the body. It's like you see the TNF alpha, oh my God, dude, why do you produce too much? It's like, I need biotin. If the baby cries, either he needs to be fed or he needs some poop, right? So the people say, oh, just crank the music. It's like, shut up, why do you bother? You have to listen to what the people, they don't want to listen. It's like, what's the purpose? Of course, over the years, this thing unfortunately turned into a multi-billion dollar business. So everybody's education. So it's
Okay. Well, good. Well, there again, acidophilus is one of my favorite ones still. It's the major one, like we talked about. That's back at that one again. I really like that one. And where I would use this is when a person has a more of a, in higher doses, not after the antibiotics so much, but if a person has a dysbiosis, in other words, they really have a messed up gut bacteria, a lot of the what we would consider bad ones growing, that's when you might want to use, the really higher doses of some of the lactobacilli, because that the dysbiosis, these will help suppress those bad guys, uh, not in the extreme way that antibiotics do, just to wipe them out, but to really put them in their place, so to speak. And that's when I think you should really start thinking about getting into the higher doses of the probiotics. L. salivarius is another one that I like well. It's especially good at suppressing. It's better than the acidophilus at suppressing the bad bacteria. It's been shown to help alleviate flatulence because a lot of times dysbiosis, you have that. Suppresses bacteria that can translocate to the pancreas and cause pancreatic necrosis, as shown in one study. Has been shown to help atopic dermatitis in children. Um, uh, I have a lot of people that like to use this after a colon cleanse. They'll take this bacteria to kind of straighten out the gut bacteria and then follow up with a more broad spectrum probiotic. Bifidobacterium bifidus is really one of the nicest ones. It's not as strong at suppressing the other bacteria. That's one you might want to put more emphasis on after the antibiotics, actually. The bifidobacteria are not going to wipe out the other bacteria to near the extent that the lactobacilli are. It's one of the most common probiotic bacteria in a healthy colon. So it's there normally. Found mostly in the colon and lower small intestine. Uh, that's the one that grows very good with inulin, the fructooligosaccharides, those kind of things. Yeah.
The slide study was the Penn State University, the same ground with inulin, but you can use it as a diagnostic tool. If you use inulin, a lot of gas and water, then you have Crohn's.
Okay.
With knowledge, you can put two negatives together and make it work.
Yeah, and a lot of things have been attributed to that. It's really one of the better ones for kind of normalizing intestinal flora. It does not stomach, it doesn't survive very well in the stomach. So you need a delivery system that will help it get to where it's going with the bifidobacteria. The lactobacillus acidophilus will survive and get through fairly well. So will the salivarius, but the bifidus doesn't. And that's why we have these delayed release capsules, or you can do an enteric coated tablet or something like that, something to help it get through the stomach before it's exposed to the stomach acid. Then, it's got a chance of working. L. reuteri is really one of the more aggressive ones at getting rid of some of those bad bacteria, more so than the acidophilus is. And this is one that you see people use for um, treating dysbiosis a lot of times. Um, in the late 1980s, Walter Dombrowskis uh, and even Casas and colleagues discovered that reuteri also produced a novel broad spectrum antibiotic substance they called um, reuterin that came as a result of its fermentation of glycerol. And it was kind of unique in that bacteria that are considered harmful were about 5 times more likely to be affected by it than the other ones. So it was really specific towards some of the more negative type bacteria. So that's a nice thing about reuteri. It's not one you want to put in a blend necessarily with other bacteria because it might kill them. So, it's better as a standalone bacteria. But it's one that can be used in the right circumstances for treating dysbiosis fairly effectively. Well, in conclusion here, the gut microbiome develops and changes throughout the life. Most microbes are introduced by bacteria into the gut that are introduced to the gut, come from the food and environment in one way or another. Um, it turns out that H. pylori is transferred from person to person only. There's not any animal vector or any other place. No other animals have it except people. So it's unique in that way. Um, very few microbes should really be considered good or bad. Uh, Even a lot of the Clostridia, which are pretty nasty, appear to have some benefits too, as long as they're held in check. Um, Most people, you hear about C. diff infections all the time. They can be very serious, but everybody in this room has that bacteria in their gut, probably. It's just that the other bacteria are holding it in place, and it may have some benefits. So, I really don't like to think of bacteria as good bacteria and bad bacteria. Just bacteria that when they're out, when they're not doing what they're supposed to do, cause problems. Um, yeah. Uh, Diet is the greatest determinant of an individual's microbiome. And this is recent, very recent Science magazine. I'll just kind of read what it says here at the very start of this article about gut microbiota. Diet affects multiple facets of human health and is inextricably linked to chronic metabolic conditions such as obesity, type 2 diabetes, and cardiovascular disease. Dietary nutrients are essential not only for human health, but also for the health and survival of the trillions of microbes that reside within the human intestines. Diet is a key component of the relationship between humans and their microbial residents. Gut microbes use ingested nutrients for fundamental biological processes, and the metabolic outputs of those processes may have important impacts on human physiology. Studies in humans and animal models are beginning to unravel the underpinnings of this relationship, and increasingly evidence suggests that it may underline some of the broader effects of diet in human health and disease. Now, that's in a review article in Science, and there again, that's Science and Nature are the two most important scientific journals that we have. So it's really rare to see that kind of stuff in science. But what they're saying is diet is fundamental to having a healthy microbiome, which is fundamental to being a healthy person. And we're still unraveling all the ins and outs of that, but we know that that's the truth. However, disease, diet, medications, they also affect the microbiome. But plant-based diets are the ideal way to establish a healthy microbiome. And that was clearly shown in two of the studies that we were talking about earlier in this talk, the one in Africa and the one in Europe, where we saw that switching to a plant-based diet has just really profound effects on the microbiomes and a positive effect. Probiotics also help, especially if a bacterial species has been eradicated. And a lot of times that's the case. That's what the antibiotics, unfortunately, sometimes do, is actually eliminate entire species of bacteria in the gut. And probiotics can replace some of those, but not all of them. But that's one thing that a probiotic can do, is replace bacteria that have been lost. We know that over the lifespan that our bacterial diversity decreases as we get my age and older, let me put it that way. So the older we get, the less of those bacteria we have, and we really have to kind of work at maintaining that bacterial diversity, because it's really important to our health. And that's where I'll stop today. And I really thank everybody who came. And if you have questions or want to discuss or argue with me, you're certainly welcome to. But it's never easy learning how to use these things skillfully and correctly. And so that's why we keep working at it and learning. And I hope, I know most people here work with people or in one way or another. So it's always beneficial to kind of hone our skills. And that's what we're trying to do. Any questions? Okay.
Very interesting.
Well, I hope you learned something. Thank you very much. I'll leave that there.
So did you come to this one and you went to the other one?
Yeah, that's what we do. And we brought